Good day, this is Professor Will, CBMD PhD, and today is Friday, November 26, 2021. It is 10:44 and 44 seconds, a.m. Eastern Standard Time. Today's research, psoriasis. So let's hop to it here. The suffering from psoriasis. Blame this trio of proteins. New study shows there may be a way to help even more psoriasis patients. Date November 19, 2021. Source: the Hoya Institute for Immunology. About 7.5 million Americans suffer from psoriasis, an autoimmune disease that shows up as patches of red inflamed skin and painful, scaly rashes. Although there are effective treatments for psoriasis, not everyone responds to these therapies, and for many, the relief is temporary. The scientists have discovered how a key protein, called TWIG, damages skin cells in psoriasis patients. Their findings in mice and with human skin cells suggest targeting TWIG may help control the disease. And these therapies don't reduce disease by 100%, and they don't cure the disease. And if you take patients off those drugs, the disease almost always comes back. And for autoimmunity and inflammation, researchers have discovered how a key protein called TWIG damages skin cells in psoriasis patients. Their findings in mice and with human skin cells suggest targeting TWIG may help control the disease. Researchers think TWIG might be considered a potential target for the treatment of psoriasis. Researchers say it is good to have this chance to develop a new therapeutic option. The findings build on the previous work showing that TWIG can interact with the most common type of skin cell, called a keratinocyte. By investigating TWIG-deficient mice, the researchers found that TWIG is a driver of inflammation in a model of psoriasis. The new study shows that TWIG does not work alone. By studying human keratinocytes, the researchers discovered that TWIG teams up with two other proteins called tumor necrosis, factor TNF, and interleukin-17, IL-17, to trigger inflammation. And this trio appears to control the production of inflammation molecules and the expression of additional inflammation associated proteins in patients with psoriasis. The fact that they work together suggests the disease is essentially driven by all three of those particular proteins at the same time. The primary implication is that TWIG will always be a good drug target, as has already been proven for TNF and IL-17. And to test this, the researchers used a mouse model of psoriasis to compare how well a TWIG inhibitor measured up to therapies inhibiting IL-17 or TNF. The results suggest the team is on the right track. If you inhibit TWIG from working on its receptor on keratinocytes, you get the same therapeutic effect as when you inhibit TNF or IL-17. And this finding is especially encouraging because TNF and IL-17 are both FDA-approved drug targets for psoriasis. Although human clinical trials remain to be done, researchers see a future for TWIG inhibitors as therapies for many types of skin diseases. Researchers think TWIG is involved in skin inflammation in general. The research laboratory is also investigating the role of TWIG in atopic dermatitis, also called eczema, a very common type of skin inflammation, especially in babies and young children. Researchers say, while psoriasis and atopic dermatitis are distinct diseases, they do have a few things in common, and there are not as many good treatments for atopic dermatitis. And there is certainly a lot of room for improvement in treatment of atopic dermatitis patients. Researchers say TWIG functions with TNF and IL-17 on keratinocytes and is a potential target for psoriasis therapy. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.